Antibiotics have become an everyday part of our lives, a simple cure to almost all infections. They are natural chemicals that are made by other kinds of bacteria and fungi that disable growth or kill bacteria. The problem, though, is that antibiotics kill both harmful and beneficial bacteria in our bodies, which can cause other health issues. The relaxed manner in which we use antibiotics has caused our bodies to slowly become resistant to them, and this issue is starting to spiral out of control. The revolution of antibiotics started with the discovery of penicillin as proven treatment for many infections, after it was experimented with on war patients and burn victims of the devastating Coconut Grove nightclub fire of 1942. From here, the use of penicillin quickly escalated into mass production and abuse, despite warning from scientists that this could cause resistant strains of bacteria. Penicillin started to be used for everything, from serious disease to drinks and soap with penicillin in them, just for preventative measures. Due to this overuse, there was soon evidence of resistance to penicillin, and by the 1950s, over half the bacteria Staphylococci were resistant. Drug resistance is most likely to occur when the drug is overused or used unnecessarily, when the drug dose given is not large enough to completely kill the infectious bacteria, when the entire course prescribed is not completed, or if a patient is on a drug for a long period of time. Antibiotic resistance happens in our microbiome, which is made up of millions of microbial cells and viruses that live in and on us. When the genes of these cells become resistant, they develop enzymes that can inactivate antibiotics by degrading or chemically modifying them. The real danger, though, happens when bacteria start to swap genes by conjugation, a direct cell-to-cell transfer, or transformation, where parts of the DNA are released by the cell and picked up by another, potentially more dangerous bacteria. This is how superbugs develop. Hospitals are hotspots for these deadly, multi-drug resistant bacteria. This becomes a serious issue as patients are treated with very strong, large-spectrum antibiotics that kill off all bacteria, leaving the body vulnerable to infection by these superbugs. Superbugs are often developed in patients with terminal illnesses or illnesses that require multiple kinds of antibiotics. The infection slowly becomes resistant to different types of antibiotics until there are none left to treat it, in which case it wins. Antibacterial products are also accelerating the resistance issue. While there has been no evidence of the benefits of adding antibacterial substances to everyday products, such as cutting boards, mattresses, high chairs, and toys, they are still being used. Antibacterials that are now used in soaps and dishwashing liquids for everyday use were once only used in hospitals for germ control. Not only does this cause resistance in bacteria that were previously present, but also causes bacteria that were not able to cause problems to now thrive without the competition of other microbes. The everyday, debatably unneeded use of antibiotics in the agricultural industry has also had an influence on the issue of drug resistance. Each year, 7 tons of antibiotics are imported into Australia, and more than half of that is used in agriculture for food and the disease prevention of animals, while only 8% of these antibiotics are actually being used to treat sick and injured animals. As we then consume the meat, which contains traces of these antibiotics, our bodies are not only being exposed to drugs when we aren't in need of them, but bacteria in our bodies are also becoming familiar with small amounts of these drugs, and most likely growing resistant to them. Eventually, they will pass this resistance on to more dangerous pathogens. Possibly one of the scariest facts I have found, the drug called avaparcin is commonly used to help the growth of cows, chickens, and pigs, but this drug is related to a last-line drug used in humans. The idea that we could slowly be becoming resistant to this last-line drug could have deadly consequences. So, do we really need to be using all these drugs in the agricultural industry? While antibiotics are used to give an advantage to animals, it has been found that they may actually be interfering with their natural stomach microbiota, and in particular a bacterium known as B theta, which plays an essential role in helping with the development and functioning of the intestines and the organizing of other bacteria. An experiment using mice with and without B theta in their bodies have found that those with it are much healthier and less susceptible to catching infection. The mice without B theta, while being fed more, put on less weight and their intestines grew irregularly. While this experiment is exaggerated, it is an insight into what we may be doing to our livestock and ourselves by having too many antibiotics. By killing off beneficial bacteria, we are risking all new kinds of illnesses that cannot be treated by antibiotics. To avoid the illnesses that we may soon be facing, we need to seriously cut back on antibiotic use in humans and animals, and also go back to normal soaps and cleaning products for everyday use. If antibiotics were used properly from the beginning, they could have been a much more beneficial solution in the long run, but consequently, they have now become an enemy.